What better way to survive the trauma of not getting Powerball lottery tickets that would fund your research for the rest of your life and a few other accessories. It is an enormous pleasure and privilege to be here today. Laura Blanton will describe her thesis work. I'm particularly pleased with our technology that we can stream this live to her many friends. I'm also particularly pleased that Mark, her dad, Susan, her mom, Catherine, her sister and best friend, Jacob, her husband's father and mother, that we're all here together to witness this journey and its punctuation point. There are many wonderful features about Laura that those of you who know her well are fully aware of. Laura has had an enormous curiosity all her life. That's evident in the shores of Lake Michigan, where she started realizing that life is complex and data sometimes are difficult to put together. She is a scholar and loves learning and sharing what she knows with others. She combines computational and experimental biology of necessity to deal with the problems that she is going to describe to you today, the scientific questions that have captivated her. I asked Laura when her parents made the religious conversion between PC and Apple, and I don't know when that was, but it's sometime after the time she was 5 years of age. Laura is generous with her spirit, her ideas, and is able to maintain a calmness and a rootedness, a centeredness, despite life's daily challenges. And I think that there's one picture that captures the look where she is able to deal with an awful lot and just to not only calm herself and to calm those around her. And I think that this view is captured by her parents when she was 8 years old?
Oh no, about 5.
5 years old, okay. Anyway, friendships, sharing, these are defining features of Laura, and I know that her best friend, her sister Catherine, is here, and how wonderful it is for her, for Laura, to have the family here, and for us to all listen to what she's going to describe to us about the role of the gut microbiota in the pathogenesis of the global health challenge, childhood undernutrition. So, thank you, Laura.
Thank you so much for that introduction, Jeff. I'm so happy to be here with you all today to tell you about the research that I've done in Jeff's lab over the past few years on the gut microbiota and childhood undernutrition. So immediately after we are born, we have our first encounter with bacteria. And over time, these bacteria come to colonize us throughout our bodies, predominantly within our gut. And this community of bacteria that come to reside in our gut, we call the gut microbiota. And in studying the gut microbiota, we've come to find that it really has many different ways in which it can shape our host biology. One way is through nutrition. So not only does the food and diets that we consume have a strong impact on which bacteria are able to colonize our gut, but in turn, the gut microbiota helps us metabolize and process lots of dietary ingredients that we consume. The gut microbiota also has heavy interactions with our immune system, helping us develop and mature our immune system. And it will interact with exogenous microbes outside of those that have colonized our bodies that might be potential pathogens and can exclude them from infecting us. And then the gut microbiota can also have physical interactions with the epithelial lining of the gut and can promote proper barrier integrity. So all of these different factors can have a healthy cycle of homeostasis when everything is going properly. But unfortunately, all of these different facets of host biology can also turn towards a diseased state, wherein if an individual becomes severely undernourished, this can lead to impaired immune function, which then can open up the host into being infected with enteropathogens, and then these enteropathogens can compromise the integrity of the gut barrier, leading to a disease called environmental enteropathy, and then that'll in turn lead back to more undernutrition. And once again, the gut microbiota is able to interact with all of these different facets of host biology. And so in looking at all of these different factors, I started thinking about the problem of childhood undernutrition, which is a devastating global health disease. So childhood undernutrition can lead to over 3 million deaths annually. And in the cases where it does not lead to mortality, there can still be long-term consequences throughout the rest of the individual's life, including stunting, that is short stature of the individual, as well as cognitive deficits. And also very importantly, many of the nutritional therapies that are being used currently are insufficient to really be able to repair or ameliorate these long-term consequences. So for my project in Jeff's lab, I decided to focus on the relationship between undernutrition and the gut microbiota. So the goal of all of this work really is to be able to have all infants and children growing healthily, whether it be through identifying individuals that are currently undernourished and getting them into a healthy state, or being even able to prevent undernutrition in the first place. And so to identify infants and children that are experiencing undernutrition, we can use definitions of anthropometry. So what that means is that the World Health Organization has created definitions of what healthy growth should look like using what they call a multi-center growth reference study, which looked at over 8,000 individuals from all over the world as these infants and children were growing over time. And they looked at metrics of these infants and children, such as their weight and their height in relationship to their age, as well as the ratio between their weight and their height. And then having all of these different measurements, they then standardized them into a Z-score so that infants and children could be compared to one another. Also indicating that if there's a particularly low Z-score, such as -3 or lower, that would indicate that the infant or child is experiencing severe undernutrition. And so we have some evidence already that the gut microbiota may play a role in promoting undernutrition, or put another way, that undernutrition is not due to food insecurity alone. And so this was work that was done by a former postdoc in the lab, Michelle Smith. And Michelle had access to a cohort of Malawian infants that were twin pairs that were either healthy or undernourished. They were discordant for their health status. And what Michelle was able to do would take these fecal microbiota from either the healthy or undernourished twin and transplant these fecal samples into notobiotic adult mice. And so what Michelle saw was that when mice received the microbiota from the healthy twin, they did not have that much change in their weight over time. But when they received the microbiota from the kwashiorkor twin, which is kwashiorkor is the form of undernutrition, she observed this profound weight loss that occurred over time. And importantly, these mice were consuming the exact same nutritionally deficient representative Malawian diet. The only difference were the bacteria that these mice were colonized with, indicating that there could be a causal role for the microbiota in promoting undernutrition. Also noteworthy was that mice colonized with this kwashiorkor microbiota also had different metabolic profiles and a decreased gut barrier integrity. And so that begs the question of what really the role of the microbiota could be in healthy human growth and development. You know, as we are born and we encounter our first bacteria, and as we grow over time, how might these bacteria be helping us grow and develop properly? And so this data came from a previous graduate student in the lab, Tanya Yatsenenko. And Tanya had access to a cross-sectional study of the fecal microbiota from 151 families from 3 different populations around the world. She was able to look at Malawians and Amerindians from Venezuela, as well as individuals from the USA. And so on this graph here, each single dot represents a single individual's microbiota, and they're plotted out by age on the x-axis here. And then what Tanya did to plot out on the y-axis was compare how each individual's microbiota was either similar or dissimilar to that from healthy reference adults from the same community, such that if there's a very high value up here, that indicates that the bacterial composition of that person's gut looked very dissimilar from that of healthy adults. But if there's a lower value down here, that indicates that the microbial composition is more similar to that of healthy adults. And what Tanya saw was that very early in life, the composition of these infants' microbiota looked very distinct from that of healthy adults. But over time, they became more and more similar to that of adults until they stabilized out at about 2 to 3 years of age, and then remained pretty stable over time after that. And so this led to the hypothesis that the gut microbiota, as it's changing over time after birth, might be playing a key role in normal postnatal growth and development. So again, as after we're born, we encounter our microbes, and these microbes seem to be developing more into adult-like state over time. They might be having an actual shaping of healthy growth and development. So to unpack this hypothesis, there are a few key questions that I had. First of which, is there a definable program of gut microbiota development shared across healthy infants and children? That is, what is the normal pattern that we would expect to see within healthy infants and children? Can we define what those patterns are? And from that, if we can define those bacterial progression patterns, what might the relationship be between microbiota development, or what we like to call maturation, and the healthy growth or nutritional status of the individual? Is there a correlational relationship? Could there be a causal relationship? And from that, if there is a causal relationship, would it be possible to identify specific bacterial taxa instrumental in promoting the healthy growth of infants and children? So to get at this first question first, I needed to be able to define what the program of these gut microbiota development is across healthy infants and children. And to be able to do that, I needed to have a way to actually measure what the bacterial composition of these individuals are. And so to do that, we can utilize a tool in the lab and in the field called 16S sequencing. And 16S sequencing works a little bit like this. So it begins by collecting the fecal samples of interest that you'd like to study. Then we can pulverize the fecal samples and extract the bacterial DNA contained therein. And once we have that DNA, we can amplify out and sequence a particular gene called the 16S ribosomal RNA gene. And this gene is very valuable because it has these alternating regions of conserved or variable regions. So the conserved regions are represented in green, the variable regions are represented in gray. And these variable regions can serve as sort of a barcode to distinguish which bacterial species that DNA came from. So all we really need is to sequence out this variable region 4 of the 16S rRNA gene. Once we have the sequences, we can group them into what we call operational taxonomic units, or OTUs. And this is done by clustering the sequences that we have based on how similar those sequences are to one another. So I use a cutoff threshold of 97% or greater sequence identity. And then once we have our OTUs, we can create our OTU counts and then really compare the differences in OTU counts between two hypothetical individuals I have here, individual A, individual B. And in this bar chart here, each color is representing a different OTU and the relative proportion of that OTU within that individual's microbiota. So now that we have ways to assess what the bacterial composition of a person's microbiota is, we needed a tool that we could use to define normal microbiota development over time. And to do this, we utilized a machine learning algorithm called random forests. So what we would do is input into this random forest algorithm our 16S sequences from the healthy infants and children from Malawi that we had access to, that we were interested in defining a microbiota maturation in. And in putting these sequences into random forests, we asked the algorithm to regress these sequences against the corresponding chronological age of that sample. And what this does, as an output, is essentially give us what we call our model of gut and microbiota maturation, which allows us to do two principal things. The first of which is that random forest is able to identify what we call age discriminatory taxa. And this indicates how informative a particular bacteria is in describing how that bacteria changes over time. So if it changes a lot over the time, it would be very age discriminatory. And because it's identified these age discriminatory taxa, the model is able to make a prediction of what the individual's age might be based solely off of the composition of the gut microbiota. So in some hypothetical examples here, we have 3 different taxa. We have taxon A in red. And random forest might look at this bacterial pattern and see that it is age discriminatory, as it's very low in abundance early on in life and then increases over time. So this would be age discriminatory. But random forest is also useful because it can pick out more complex patterns than just the simple one in taxon A, and more complex patterns like in taxon B, where it's low in abundance early in life, peaks at a certain point, and then decreases again. So taxon B in blue would also be age discriminatory. There may also be taxa, like taxon C in green, that are not so age discriminatory or not so informative for the model, as it doesn't really change over time. And so to refine this model, I run iterative regressions of it because it's not a deterministic model. And through all of these iterations, we can build a consensus ranking of how age discriminatory each of these taxa are. And because we've ranked all of these taxa by how informative they are to the model, I can refine the model even further by trimming away variables or OTUs that aren't so discriminatory. So I had access to a healthy cohort of Malawian infants that I was able to use to define normal postnatal microbiota development. And these 2 histograms represent the way in which I subsetted out this healthy cohort to build my model. So this histogram on the left here represents how many samples were in each age bin from 0 months to 32 months. And these were the samples that I subsetted out to train the model initially. While this histogram in gray represents the samples from my healthy cohort that were excluded from model training that I kept out so that they could be used to validate the model later. So I call this black histogram here my training subset versus my testing subset here. So now that I've got these healthy cohorts and I've regressed them against chronological age and random forests, I wanted to see actually what my most age discriminatory taxa were. So this is what we're looking at here. This axis here is called the increase in mean squared error, and this represents how much error would be introduced into the model if that bacteria taxa were not there. So they're ranked by increase in mean squared error because if there's more error introduced into the model in the absence of that taxa, that means that taxa is very important. So at the top of the list we have our Bifidobacterium longum, which is not that surprising to us because Bifidobacteria is a species that we see frequently within infants. And another thing that we noticed looking at our list of most age discriminatory taxa within my Malawian cohort is that there were some taxa that were also found to be age discriminatory when we did the same sort of microbiota model maturation within a Bangladeshi healthy cohort. So this showed us that there were taxa that were shown to have global patterns and changing over time that were shared between both Malawi and Bangladesh. And so this is the same list of the same 25 age discriminatory taxa that I showed you on the previous slide, but this is showing the relative abundances over time. So each row represents a single OTU, and then along here by the columns, they're binned by the ages in months. So we have 0 months over here and 30 months at the far right. And we can see very early on in life, there's a high relative abundance of many of these bifidobacteria here, which is again what we would expect. But over time, these bifidobacteria decrease in the relative abundance until we can't really detect them anymore. And this whole other large swath of bacteria come in that are highly diverse. These include a lot more firmicutes. We see Faecalibacterium prausnitzii, Clostridia, and other species like that. So now that I've identified these bacterial taxa that seem to be age discriminatory, I wanted to validate my model to see how well it actually is able to relate to chronological age. So that's what we're looking at here. So I mentioned before the subset of my healthy cohort that was excluded from model training, my test set, and so now I'm using my test set to validate how well my model performs. So these samples represent the samples from my test set. The chronological age of their samples is plotted out on the x-axis, while their predicted microbiota age is plotted out on the y. So this is the output of the model, only looking at the bacterial composition of these samples and making a prediction about what the age of these samples are. And what you can see is that there is a strong positive correlation between the actual chronological age of these samples and the predicted microbiota age. I was able to further validate this model with a separate cohort of Malawian healthy singletons. And these were samples that, these were individuals that were only sampled at 6, 12, or 18 months of age. And again, we have their chronological age here on the x, their predicted microbiota age in months on the y. And again, I was able to see this strong positive correlation between the actual and predicted ages of these individuals. So I was able to validate my model, but I wanted to go one step further in the analysis of these microbiota ages. And so to do that, I created a microbiota for age z-score. So this is a z-score analogous to the anthropometric measurements created by the WHO that I mentioned at the beginning of my talk. And this was done by binning the different microbiota ages that we saw by months over time in these different individuals. And then within each age bin, calculating the median and standard deviation, such that if there is an individual whose predicted microbiota age is right at the median, they would have an MAZ score, as we would like to say, of right around 0. But if an individual has a microbiota age that is far below the median, like in this pink dot here, this might be around an MAZ score of -2 or so. And having created this MAZ score as a metric for looking at an individual's microbiota maturity, I then wanted to ask if there is a difference in the MAZ scores between healthy and undernourished children. And so that's what we're looking at here. So these are 18-month-old Malawian singletons. And I was able to see, looking at the MAZ scores of either healthy or undernourished children, that infants that were anthropometrically undernourished had significantly lower MAZ scores compared to their healthy age-matched counterparts. So even though these singleton Malawian infants were the same chronological age, the infants that were experiencing undernutrition had significantly younger-looking microbiota than what we would have expected given their age. So in this first part of the talk, I talked to you about this model of microbiota maturation, where we saw that healthy individuals have this predictable pattern of bacterial patterns that change over time, this microbiota maturation, such that we're able to distinguish between healthy individuals by their younger looking or older looking bacterial compositions. And then I also showed you this evidence that of the difference in microbiota maturity depending on health status, such that these undernourished children have microbiota that look significantly immature compared to their healthy age-matched counterparts, such that even though these individuals may be the same chronological age, infants that are undernourished have microbiota that look younger than what we would expect. So having been able to define this program of gut microbiota development over time, I then wanted to ask what the relationship might be between this microbiota development or maturation and actually the healthy growth or nutritional status of these individuals. And so to do that, I turned to our notobiotic mouse models within the lab. And so here I've got pictured our two notobiotic facility directors, Maria Carlson and Dave O'Donnell, without whom none of this would have been possible. And you can see here on the left, this is a picture of our notobiotic or germ-free mouse facility, and you can see the different vinyl isolators here going down the rows. And utilizing these notobiotic mouse models is extremely powerful for a number of reasons. So what we'll do is take an individual's fecal sample, transplant them into germ-free mice. So these are mice that have never experienced any bacteria previously in their life. But we can set up experiments that include replication, so giving the same microbiota to many different mice. We can highly control all the environmental conditions within these experiments, including the diet that these mice consume. So we can manipulate the diet to determine the effects of the dietary ingredients on the composition of the microbiota. And this really allows us to ascertain which phenotypes are attributable to the gut microbiota. And so in designing these experiments, I really tried to replicate three principal things. One, the microbiota, which we have from the individuals from Malawi, the growing host, which is through these nootropic mice, and then a representative diet that replicates or tries to represent the diet consumed by the individuals that we want to study. So I was able to develop this representative Malawian diet, which we like to call M8. This is our collaborator, Kay Dewey, who was in charge of conducting this dietary survey of the complementary feeding patterns of Malawian infants and children enrolled in this clinical study called Islands Dose. And in this dietary survey, they looked at both the actual foods that these infants were consuming, as well as the nutritional components of these diets. So this is the composition of the diet that we ended up designing. You can see it's predominantly corn flour, which is about a quarter of it, so it's a carbohydrate-rich diet. And this diet is also nutritionally deficient. So this represents the different macro and micronutrients of this diet. And then here we have the percent of how much the daily mouse's nutritional requirement is fulfilled by this diet. And this mimics the daily nutrition that the Malawian infants receive. And so we wanted to mimic that as much as possible. So you can see that this diet is deficient in protein, a number of B vitamins, etc. So we have all the tools that we need to conduct this experiment. And this experiment that we wanted to do really wanted to ask whether the gut microbiota may have a causal effect on host growth. So I did this in strong collaboration with Mark Charbonneau. And what Mark and I did was identify a number of human donors that we had the fecal microbiota of. And these were 6 or 18-month-old donors that were either healthy for anthropometry or undernourished. For each fecal sample of these individuals, we transplanted them into 5 different mice, and these were mice that were recently weaned germ-free mice. We did this when the mice were 5 weeks of age. And then after they were colonized with their fecal microbiota, we simply followed them over time for 5 weeks afterwards until sacrifice. And over this 5-week period, we assessed their growth. We did this through total body weight by simply weighing them, as well as looking at the body composition of these mice through a quantitative MRI, or QMR. And this allowed us to measure the lean mass of these mice. And when we were developing these experiments, it also occurred to us that another parameter that we might want to look at is the bone morphology of these mice, as we're interested in studying proper growth and development, and bone could be another facet of that. So what did we see? We saw that both by total body weight and lean mass gain, mice that had been colonized with a healthy individual's microbiota grew significantly more than those mice that had been colonized with an undernourished individual's microbiota. So we have in time here on the x-axis, while we have the percent of initial weight or percent of initial lean mass gain here on the y, and the individuals that were colonized with the healthy microbiota in blue, and the individuals colonized with the undernourished microbiota in red. So this suggested to us that there might be a causal role of the gut microbiota in healthy growth and development, particularly because we also observed that there was no difference in food consumption between these treatment groups. So it wasn't simply that the mice growing more were eating more. Another interesting observation from this was that we saw that actually microbiota from 6-month-old donors conferred greater growth than 18-month-old donors. But however, within each age bin, regardless of whether you were from a 6-month-old or 18-month-old microbiota, the mice colonized with healthy derived microbiota conferred more growth than undernourished. So I mentioned that we also looked at some bone morphology of these mice. So what we did is we dissected out the femur. So this is a schematic of the anatomy of the femur. We have the proximal here, this would be towards the hip, versus the distal here by the knee. This midsection is called the cortical region, while we have the spongy area, or the trabecular region of the bone here. And so we wanted to compare if there are any differences in bone morphology, depending on whether the mice had been colonized with a healthy or undernourished donor. We saw trends towards differences between the healthy donor and undernourished donor recipient mice in cortical bone volume ratio to tissue volume here, as well as in this cortical region in the volumetric bone mineral density. We also compared whether the mice had been colonized with a young versus older microbiota, and here we did see significant differences, such that the mice that had been colonized with the 6-month-old donor microbiota had significantly higher trabecular bone volume to tissue volume ratio, as well as trabecular bone mineral density. So we've observed these differences in phenotypes depending on whether the mice have been colonized with a healthy or undernourished individual's microbiota, but we wanted to be able to harness all of this data to really generate hypotheses about which specific taxa in these microbiota may be conferring differences in growth. And so to do this, we turned again to random forests, and then this time, instead of regressing these OTUs against chronological age like we had done initially, we regressed the OTUs against the growth phenotypes of the mice. And in this allowed us to identify growth discriminatory taxa. So here, this is a list of the taxa that are the most weight discriminatory. But you'll see that a number of these taxa are highlighted in red. And these indicate taxa that were both found to be weight and lean mass gain discriminatory. And so looking at this list, you can see some patterns. For example, there are many OTUs that are popping up from the species that are assigned to the species R. navis. And also, I was curious as to how many of these taxa might be having patterns against age within our human cohort. So I took these same OTUs and looked at their patterns over time in age within our healthy infant Malawian human cohort. And the OTUs with stars next to them are OTUs that I also found have a significant correlation to chronological age in the healthy Malawian cohort that I used to model gut microbiota maturation. So in this part of the talk, I showed you that we have some evidence that there may be a causal role of the gut microbiota in post-growth, such that mice that received microbiota from healthy infants and children conferred greater weight and lean mass gain to these recipient notobiotic mice than the mice that had received microbiota from undernourished individuals. So moving on to the remaining question, now that we've seen that there can be this causal relationship between the health status of the individual and healthy growth and growth of the mice, we wanted to drill down to see if we could identify specific bacterial taxa that are able to promote healthy growth. So to do that, and to rephrase that question, are there microbes that from a healthy microbiota or from a healthy derived individual microbiota that are able to promote this growth? And so to do this, I identified 2 groups of mice that had highly discriminate growth within my initial screen experiment. So this indicates the days post-colonization, so over time, how these mice were growing. And this is the lean mass gain of these mice. So this was a group of mice that received the microbiota from a healthy individual that grew a lot. And this is the group of mice from an undernourished individual that did not grow so much. You can see the anthropometry of these individuals here. This is the healthy versus undernourished individual. And you can see that this undernourished individual is severely underweight. This is the weight for HZ score, as well as severely stunted for the height for HZ score. You can also see the predicted microbiota ages of these individuals, such that the healthy individual, both of these individuals, I should say, are 6 months old at the time of sampling. This person's microbiota age was 6.8 predicted months, compared to the undernourished individual, which had a slightly younger looking microbiota of 4.6 months. And these 2 different communities were also highly distinct from one another, which is what's displayed over here in this bar chart. We have the healthy individual's microbiota on the left here. And looking at this healthy individual's microbiota, I noticed that there was a high dominance of F. prausnitzii, which is in this large blue section here, as well as a high proportion of R. navis. And these were interesting to me because both R. navis and F. prausnitzii were found to be highly weight discriminatory within my random forest modeling. While in contrast, the undernourished community did not have any F. prausnitzii or R. navis. Instead, it was highly dominated by this Clostridium neonatale here, and as well had a little bit of B. longum, which is represented at the very top there. So now that I've identified these 2 highly discriminatory groups of microbiota, what am I going to do with them? So One way that I wanted to see if I could use the healthy microbiota to influence the undernourished microbiota was an experiment we call a co-housing experiment. This utilizes the natural behavior of mice in that they are coprophagic, meaning that they'll eat one another's feces if they are occupying the same cage. And so the design of this experiment was as follows. I had a group of mice that were colonized with a healthy human microbiota, represented in blue here. And then I had another group of mice that were colonized with the undernourished microbiota, represented in red. After a period of time, I took a subset of these mice, each of the healthy recipient mice or the undernourished recipient mice, and then co-housed them within the same cage so that they would be able to be exposed to one another's microbiota. And then as controls, I withheld mice from each of these groups in either my healthy controls, which is the very top arrow, or my undernourished controls, which is the very bottom arrow. And these were controls because these were mice that were never exposed to a different microbial community than their own. These mice were also colonized at 5 weeks of age, like in my previous experiment. Then 4 days after co-housing, 4 days after colonization, these mice were co-housed. And then I followed the growth of these mice for a total of 21 days after their initial colonization before sacrifice. So this is just another schematic of the same experimental design where you can really appreciate the outline of the isolators within the germ-free facility that we had to have for this experiment, such that initially there are 2 different isolators, isolator 1 and 2 on the far left. And within these isolators were mice that were colonized with either the healthy or undernourished microbiota. And then 4 days after these mice were colonized, a subset of mice from the first 2 isolators were brought into a third isolator, isolator 3, and these mice were dually housed so that within each cage there would be one mouse originally colonized with the healthy microbiota, which is the HCH, and another mouse that was originally colonized with the undernourished microbiota, which is the UNCH. So in comparing the different groups from this experiment, there'll be 4 different groups of mice that we're looking at. Either the healthy control, the undernourished control, the mice originally colonized with healthy microbiota and then co-housed, and then vice versa, the mice originally colonized with the undernourished microbiota and then co-housed. So what happened to actually the growth of these mice? Well, this is a representation of the lean mass gain of these mice. These is my undernourished control here on the left, and these were the mice that were my undernourished and then co-housed mice. And I saw that there was a significant increase in the lean mass gain of the mice that originally colonized with the undernourished microbiota and then co-housed. So something must have happened in the process of co-housing to increase the growth of these mice. And then in contrast, when I compared my co-housed mice to my healthy control, there was no significant difference in the lean mass gain between the between the growth of the healthy recipient mice and the co-housed mice. So we've seen these growth phenotypes. We also wanted to look at what was happening at a microbial level on the dynamics in the midst of co-housing. So that's what these heat maps are representing here, such that each row across the way represents a single species that was present within the experiment, while each column in these heat maps represents a single mouse on a given day, and they're grouped by experimental group. So on the far left cluster here, we have all the mice within my healthy control that were only ever exposed to the healthy individualist microbiota. And then it's moving over time from left to right. And then in this righter cluster of heat map here, these is my undernourished control mice, such that they were only ever exposed to the undernourished microbiota. And you can see the large differences in the microbes that are present in each community, such that there's a whole swath of bacteria highly abundant within the healthy community that are completely absent within the undernourished, and then vice versa. So these were our controls. They looked just as we expected. What happened within our experimental groups? So this is the group of mice that were healthy co-housed mice, with the time of co-housing delineated by this arrow and white bar right here. And you can see that even after co-housing, there's not much difference between the microbial levels of these mice, even though they've been exposed to the undernourished recipient mice's microbiota. In contrast, looking at the mice that were initially colonized with the undernourished microbiota and then co-housed, we can see before co-housing, they look similar to that of the undernourished control as we would expect. However, after co-housing, there was a large influx of many of the bacteria from the healthy derived microbiota into the undernourished co-housed mice. And in tandem with that, there was a large decrease in the bacteria that were previously highly abundant within these mice, such to below the level, such that they were displaced below the level of detection. So there are these two different clusters of bacteria that we kind of delineated here. The top in the blue box here that came in that invaded the guts of the undernourished co-housed mice, and then within the red box, a whole group of bacteria that decreased their abundance after co-housing. And so this begs the question, what has led to this increase in lean mass gain that we saw, that I showed on the previous slide? Is it this invasion of healthy associated taxa within the blue box, or is it the decrease in the relative abundance of these undernourished associated taxa represented within the red box? And looking in this further, we were also interested because we noticed many of the taxa that were present within this blue box that I've starred here with asterisks, we also found to be weight discriminatory in my previous random forest model that I showed you earlier, including taxa such as the R. mavis, which we saw many OTUs of, S. brasnitzii, and other firmicute species. So having seen this, we wanted to really tackle this question more directly of what's leading to this increase in lean mass gain. And to do that, I collaborated with another graduate student in the lab, Tarek Salih, to really be able to culture out and isolate what we're calling these growth discriminatory invaders, to see if we can culture out these taxa and then conduct specific experiments with them. And so to do that, this is the pipeline that we had here, which began with our fecal sample interest from the infant. We then would bring these fecal samples into our anaerobic environments, these koi chambers that we have within the lab, grow them up, dilute them, plate the bacterial suspensions on rich media, and we can pick the specific colonies from this suspension such that each colony represents a single bacterial cell to begin with. So these are pure cultures. We can colony up and to grow single isolates, and then once we have a whole group of isolates, we can identify the actual taxonomies of the bacteria that we've been able to culture through 16S sequencing. So what did we get through this initiative? Well, we were able to culture out 5 different taxa that were of interest to us because they fulfilled these different parameters here. All of 5 of these taxa we found to be either growth discriminatory or correlated, as by the random forest model. All 5 of these taxa also were able to invade within my co-housing experiment. And also 3 of these taxa, F. brasnitzii, Doria generans, and Clostridium symbiosum, were either age discriminatory or correlated with age within the healthy human Malawian cohort. So since we've been able to culture out this consortium of 5 bacterial taxa, we wanted to directly ask whether these growth discriminatory isolates might be sufficient to prevent the growth faltering produced by the undernourished, which is what the UN here means, the undernourished donors microbiota. So in this experiment, I simply took mice colonized with the undernourished microbiota, and within the same gavage that these mice received, or within the same colonization event, we spiked in the 5-member consortium such that mice were colonized with both the undernourished donor microbiota and our growth discriminatory 5-member consortium all at the same time. And we simply wanted to ask if there'd be a change in growth as a result of this changed, altered microbial composition. Once again, these mice were colonized at 5 weeks of age. They had been recently weaned, and we followed them for 21 days after colonization before sacrifice. So we took a look at which of these bacteria actually colonized. So on the left, I have my undernourished control. So these were mice that had not been exposed to these growth discriminatory isolates. While on the right, I have the mice that were exposed to both the undernourished microbiota, donors microbiota, as well as the 5-member consortium. And we saw that all of these 5 different taxa, only the Ruminococcus navis and the Clostridium symbiosum were able to successfully colonize the undernourished donors microbiota in this context. So you can see here with the arrows, the Ruminococcus navis is represented by that sort of large pink block at the top of the bar graph. It ended up taking about 20% of the community, while the Clostridium symbiosum is represented by the purple block at the very top, and the C. symbiosum ended up taking about 3% of the community. So what actually happened to the growth of these mice? We saw that the addition of these healthy associated taxa was in fact sufficient to enhance the growth of these mice. So we have total body weight on the left, lean mass gain on the right, and you can see that there's a significant increase in the total body weight gain and lean mass gain of the mice represented by the gray bars, the mice that received both the undernourished donors community and the growth discriminatory taxa. So in this section, I described to you how we've been able to alter the composition of the gut microbiota and subsequently alter the host phenotypes. We did this two ways. First, through the co-housing experiment, where we co-housed together mice that had been either colonized with the undernourished donors microbiota or the healthy microbiota. And we saw that in the process of this co-housing, there was robust invasion of many of the taxa from the healthy derived community into the undernourished community, and there was this concomitant increase in lean mass gain in these mice. We also then more directly tested the change in altering the gut microbiota and changing in host phenotypes through the direct addition of R. navis and C. symbiosum into the undernourished community, and saw that there was this increase in weight and lean mass gain. So in summary of the three main questions that I've described to you today, I told you about how I've been able to develop and validate this random forest-based model of what we like to call the maturation of the gut microbiota based on healthy infants from Malawi. And in addition to be able to define these bacterial progressions over time within healthy individuals, I was also able to see differences in the microbiota maturity of infants that were experiencing undernutrition compared to that of healthy children. I also told you about the notobiotic experiments where I was able to see that microbiota from healthy infants and children conferred greater growth into recipient mice than mice that were harboring microbiota from undernourished infants and children. And these types of experiments really have provided preclinical evidence that the gut microbiota and its immaturity might be causally related to impaired growth. And we also were able to see that the age discriminatory taxa that I was able to identify might not just be biomarkers of gut microbiota development, but a subset of these taxa are also effectors of host growth. So we've seen these phenotypes on the microbiota that are impacting host growth. These differences in whether the mice are colonized with the undernourished donors microbiota or the healthy donors microbiota, and the changes in weight and lean mass gain, which are not attributable to differences in food consumption. And from here, we really want to figure out what might be going on. And because of these differences in growth phenotypes, it occurred to us that there ought to be changes in the host metabolism to manifest these changes in phenotypes. So we wanted to ask how host metabolism might be changing depending on the composition of the microbiota. So we took a look at the host metabolic profiles of mice that were harboring either my healthy donors microbiota or my undernourished donors microbiota. And one of the things that we saw was that there were differences in levels of amino acids within the gastrocnemius muscle. So this is sort of equivalent to the calf of the mouse, such that mice that were colonized with the healthy donors microbiota had significantly lower concentrations of a number of amino acids, particularly we were interested in seeing these branched chain amino acids here, that they were lower in the healthy donor recipient mice than the undernourished donor recipient mice. And I also noticed that there were changes in these acylcarnitines, which are intermediates of fatty acid metabolism, in both the serum and the livers of these mice. But these were reversed differences in concentration, such that there were higher levels of these acylcarnitines in healthy donor recipient mice compared to undernourished donor recipient mice. So these differences in the metabolic profiles of these two groups of mice indicated to us that there might be some sort of difference in how these mice are utilizing fuel, depending on whether they're colonized with the healthy or undernourished microbiota. Similarly, we took a look at the metabolic profiles of the mice that had been either colonized with the undernourished donor microbiota or supplemented with the R. navis and C. symbiosum growth promoting taxa. And so this heat map represents the differences in the metabolic profiles that we saw between these two groups of mice. And of these metabolites, we were particularly interested in these acylcarnitines that were different within liver. So these shorter chain acylcarnitines are acylcarnitines that can often be indicative of branched chain amino acid, and we saw that there were higher concentrations of these liver acylcarnitines in the mice that were colonized with the undernourished donors microbiota compared to those that also had the RNA-vis and C-symbiosin within their gut. So this leads us to the potential hypothesis that mice harboring either the microbiota from the healthy donor or the growth-promoting taxa might be having differences in their utilization of amino acid pools, such that they may be funneling these amino acids into protein synthesis or lean mass synthesis, whereas those that are not harboring the healthy taxa, such as the mice harboring the undernourished donor's microbiota, might be preserving these amino acid pools for actual energy production. But there's lots more that we really need to flesh out about the metabolic dynamics of these two different groups of mice. This is a schematic from Deb Mule et al. That represents how there might be changes in the flexibility of the metabolism of different donors and how important it is to really make sure whether the host is efficiently utilizing fuel or not. So there's this concept of metabolic flexibility, which is sort of the proper healthy state here, which is represented here, such that soon after a meal, there's high utilization of carbohydrates, but over time, as nutritional cues change and there's less food available, and one turns to fasting, there's then a shift towards utilizing more fats. Well, improper, inefficient use of fuel, or is the case of metabolic inflexibility represented here, where there's sort of a mix of both the utilization of carbohydrates and fat at the same time. And so this phenomenon occurred to us in looking at the metabolic profiles of my mice, and we're interested in determining how these mice might be responding to either the fed or fasted state. In particular, because all of the metabolic profiles that I showed you on the previous slide are all a reflection of mice within the fed state. So we don't really know what's happening and how these mice might be responding to changes in their nutritional cues. This is all the host side that we still have yet to characterize. But we're also interested in characterizing what might be happening on a microbial level. So this is a dense map that you're not intended to fully be able to read, and that's on purpose because it reflects the complex metabolic interactions that can happen within microbes at a time. So this is a metabolic reconstruction that we can create of taxa within the gut. This can give us clues about what each microbe is able to contribute to the host environment and might be changing depending on what the microbial composition of the host is. And from here, this is all toward the effort of trying to identify new therapies to try and address this global problem of childhood undernutrition, but the therapies that might be keeping in mind the microbiota. So I like to call these microbiota-directed therapeutic foods, such that we might be able to either repair or prevent undernutrition or microbiota immaturity in the first place. So I was able to show you some aspects of this cycle that we're working through throughout my talk today. So on the left circle here, you can see how we've been able to leverage existing clinical trials and bring them into experiments with notobiotic animals so that we can really identify taxa that we think might be effector taxa and understanding the phenotypes and responses that are happening as a result of different bacteria within the gut. But we'd like to move into more of this right cycle here, where in studying the microbes that we found to be interesting, we might be able to identify what we like to call prebiotics or probiotics, the combination they're in, or symbiotics, such that things might be therapies to enhance the representation of beneficial taxa within an individual's gut to try and repair microbiota immaturity. And in identifying these prebiotics, this then might be able to lead us towards clinical trials that might be more microbiota directed. And if we're able to really implement durable repair of microbiota immaturity within these individuals, we can then ask whether this has an impact on clinical outcomes. So with that, I would like to acknowledge everyone that has been instrumental to this work. First of all, I would, of course, like to thank Jeff, who's just been a phenomenal mentor, and I am so grateful that I've been able to join his lab and get the opportunity to do this type of project, which has been so exciting throughout the entire way. I also would like to acknowledge everyone on my thesis committee who's been so helpful with all of their feedback and all of their time, recognizing multiple different sources of funding. And I particularly like to recognize the people instrumental in the molecular genetics and genomics program, Tim and Jim, and of course Melanie. I would like to thank everyone in my lab that has been so supportive and so helpful throughout all of this work. Sabrina, Dave and Maria, Justin, Marty, Janaki, Sue, and Michael Barrett have been incredible infrastructure support throughout not just my project, everyone's project. And I'd also like to thank Jess Hoisington-Lopez, who is our sequencing guru, and we would not have any data without. I worked closely with Mark Charbonneau and Tarek Salib throughout the course of this work, and I'm very grateful for them for all of their assistance and support and everything, and just being wonderful lab partners. I'm also very grateful to everyone in my lab, because we're not just a bunch of coworkers, we're a community of friends, and I've had wonderful times with them over the years, whether it's Sabrina teaching me to knit, that's the photo in the middle, or different celebratory dinners on the upper left, accidental matching outfits in the upper right, pig roasts on the left for just general silliness. So I'm really grateful to have been in such a wonderful lab that is just an excellent community. I would also like to thank everyone that I worked with in the Young Scientist program. YSP ended up being an integral part of my graduate student experience that helped me keep me balanced with my research, helped me stay engaged within the community. And so I'm really thankful to all of my friends that I've developed there. I was co-director of YSP with Molly Gibson, who's able to be here today. And I've worked also closely with Reka and Bo and so many other people that I don't have time to name, but I'm just grateful for all of them and their dedication to education. I have many fantastic friends that I have been able to either meet in St. Louis or stay in touch with while I'm out here. In the upper left, there's a photo of all the people from Haverford. There's many pictures up here of people from graduate school that have become dear friends. Other people that Jacob and I have been able to get to know through Second Prez or his own co-workers and things like that. So I just want to thank everyone up here as well for being such supportive companions throughout all of my time here. And of course, I would like to thank my family. So I wanted to put some embarrassing pictures of not just me up during this whole presentation. So I'm very thankful to have such a phenomenal family. My mom and dad, and then Jacob's parents as well, who are with us today, are such supportive people, and they've lifted me up throughout my entire graduate school process, and I'm grateful to them. My sister has always shown me the way of the world. She's been happy to tell me like it is, and so this is her telling me what's going on with this book even here. And then on this upper right photo here, that's my sister and what would end up being her husband many years later. So here we are present day, the second picture in from the right, with their daughter Claire, and I just love them so dearly. I'm so glad that they're here with us. And one day, for a wedding, the photo up in the upper left is my mom, and we accidentally had matching outfits, and so that was embarrassing as well. And I also have some photos of my grandparents up here as well, who I love very dearly. Lastly, I would like to thank my husband Jacob, who is the best dance partner I could have ever imagined. But even off the dance floor, he is the best life partner ever. And so thank you to him as well. And with that, I'd be happy to take any questions. Thank you.
Are there questions from the audience?
Can you elaborate on the idea of the unhealthy microbiome being younger, especially how you tell the difference between younger and just different?
All right, so the idea of the microbiota looking younger specifically comes from the looking at the age discriminatory taxa that we identified and the relative abundances of them. So when we've looked at the composition of these individuals, within the model, it's only taking into account those 25 taxa that I described earlier. And so in looking at all of the different patterns of those taxa within an individual, it comes up as looking younger if it resembles that of the younger healthy infant. And so when it comes to the difference between whether it looks younger or different is a bit tricky to tease apart. I can't really think of an example in which I've encountered a microbiota that just looked different, and that, there was a good example of that. So I'm not really sure how to get at that part of it. But that's what it means when I'm trying to describe a microbiota that looks younger or immature than what we would expect. Yeah.
Do you think the effect that you've seen by the microbiota on bone growth is, and that you've implicated for weight gain, do you think those same amino acids that you've implicated are causal in that are also causal for the bone growth, or are they independent?
Yes. So let me rephrase your question. So so you're trying to compare the differences in bone growth versus the differences in metabolic profiles. So the strongest phenotypes we saw in the femoral bone morphology that were really intriguing were between that of the mice that have been colonized with either the 6-month-old or the 18-month-old microbiota. And I haven't been able to compare the metabolic profiles of mice that were colonized with either the 6 or 18-month-old microbiota. So I can't really match up those differences there. So it's not a complete comparison that I can really say.
So I guess most of the work is essentially based on relative abundances of the microbial community, right? Is there any reason to believe that part of undernourishment might have something to do with the total gut microbial content rather than the relative abundances?
And so it's sort of the total biomass of how many microbes are there. It absolutely could be. We haven't looked into that. I think we haven't come up with the best way to really assess how much biomass is present within an individual microbiota. But there's definitely, that's definitely another valuable parameter to think about going forward.
Well, not seeing any more hands lifted to the sky. I thank everybody for your attention, and we will be having a gathering around 4 o'clock on the 4th floor of the new 4515 building, and we look forward to having you attend if you're free. So thank you very much, Laura.